Welcome to this session entitled E. coli serotype identification, serotype finder tool description and application. My name is Rolf Sommer-Kaas. I'm a bioinformatician with the National Food Institute here at the Technical University of Denmark. In this session, I'm going to talk about serotype finder, which is a tool to serotype E. coli from sequencing data. Now, I'm going to start out by speaking a little about the method behind serotype finder. This will be very briefly, and if you want more details, you should consult the publication by Katrine Jonsen, and this also contains the validation data. Then I'm going to speak a little about how to run the web tool, and I'll show you some of the output. Now, when we do serotyping using whole genome sequencing data or sequence data in general, we can employ two different strategies. We can either use direct targets, where we are trying to locate the genes that are actually encoding the phenotype, or we can try to look for indirect targets, which is markers that we expect will follow the serotype. Now, serotype finder are using direct targets. And specifically, it's using W, C, X, Y, M, and T genes for the O type, and then it mainly uses the fly-C gene, and for a few types, it uses non-fly-C genes listed here. Now, a serotype finder is an assembly-based tool, which means that even if you provide raw data to the tool, it will use the CGE assembler to assemble your raw data into context, which it will then feed into the actual tool, which blasts your context against the gene databases. And of course, if you provide assembled data, it will just blast your context against the gene databases. So the database is a curated database. It's curated by Flemming Schøjts at the Danish Public Health Institute, SSI. And currently it covers the O types 1 through 187, with the exception of O14 and O57, and it covers all 53 H types. There is some discrepancies between the genotypes and the phenotypes, and this is mainly to do with the phenotypes that is typed as rough, non-motile, or non-typable. These will often get a genotype that is an actual serotype. Now, serotype finder does have some issues, and the issues is with the types that is listed down here. And the issue is that the genes that we are using to actually serotype are completely identical for these different types, and therefore we cannot distinguish the different types. So serotype finder is found at this address, and the front page looks something like this. The tool was initially designed to hold several databases, but at the moment it only holds the E. coli one. So there is actually only 3 options that you need to be concerned about. So the first one is the threshold for percentage of identification. So if you have a gene and your contact, and they have been aligned by BLAST, then you have the alignment region here, and what this tells you is that at least 85% of the nucleotides in this alignment region needs to be identical. Then the next option speaks about the length of this alignment region. So here it says that at least this region must be at least 60% of either of these two of the genes. Sorry. And finally, you also have to specify the type of data that you put in. And then you upload, and you will get an output that looks something like this. So you will first of all get a table, and the table tells you which gene has been found. It tells you the gene name, the percent identity of the gene and in the alignment, and the length of the gene that you have. And then it will tell you the name of the contact, where the gene was found, and the position in those contacts. And then it will also tell you which type that the specific gene encodes for. And you can get an accession number here, which is a link you can actually press so you can get the data for the specific gene in the database. The actual predicted serotype is then below the table here, and you can push the button here called Extended Output, which gives you a view of the actual alignment done by BLAST. Then you can download your results as text or tabulated as text file, which can be imported to Excel or another spreadsheet software. And then you can download the specific sequences for either the gene or the hit in the gene. And the last 3 lines here basically just gives you information of the settings that you have used, your percentage threshold, your minimum length, and which files that you put into the tool. Finally, I'm just going to note that most of the CGE tools are assembly-based, and this means that even if you provide them with raw data, then the raw data will go into the CGE assembler, which then again will create context that it will feed into the actual tool that you're using. Therefore, you can optimize your workflow by just doing one assembly with the CGE assembler or the CGE pipeline, and then submit your assembly to the various tools that you want to use. Thank you for watching.